Hello, I'm Professor Sims, and in this video I will discuss ubiquity of microorganisms, culturing and isolating bacterial microbial colonies. This is the second in the series of 10 lab sessions held as part of my online Laboratory for the Fundamentals of Microbiology course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include learning to perform proper aseptic technique, understanding the meaning and significance of microbes being ubiquitous in nature, learning about different physical types of media like liquid broths, solid agars, slants, deeps, and plates, becoming competent preparing streak plates and isolating bacterial colonies, being able to observe and describe microbial colony morphologies, and as always, understanding safety and disposal procedures relating to this lab. Microorganisms are ubiquitous in the environment, which means that they are found everywhere. One characteristic of microorganisms that contributes to their ubiquity is the fact that they are very diverse. There are three major classes of microbes, which are going to be your prokaryotes, eukaryotes, and viruses. Prokaryotes are rather simple in their structure. They lack membrane-bound nuclei. They don't have membrane-bound organelles. They're usually unicellular with a cell membrane, ribosomes, and chromosomes. Eukaryotes are a bit more complex and they're usually much larger. They have a membrane-bound nucleus and organelles. They can be unicellular or multicellular. They also have cell membrane. They have complex cell structures like your mitochondria, Golgi apparatus, that kind of thing. Viruses are really quite different from bacteria in that they have a very simple structure. It's basically just genetic material surrounded by a protein and lipid membrane, and they are dependent upon a host or the host's infected cells in order to multiply and disperse. Another characteristic of microorganisms that contributes to their ubiquity is the fact that they are very, very old. They've had up to 4 billion years to evolve and to adapt to live in basically any and all niches. The fossil record shows bacteria have existed on Earth in some form or another for about 3.5 billion years. So here we have the estimated origin of the Earth. And right away, you've got prokaryotes. Note, too, that eukaryotes came much, much later than prokaryotes. The first experiment for Lab 2 is going to be twofold, right? So you have the learning objectives for this one are going to be illustrating the ubiquity of microbes in your environment. And then you're also, again, going to be modeling the scientific method because you want to formulate and test a hypothesis. So what you're going to do is you're going to choose 2 inanimate objects in your home to test for bacterial contamination. So some examples might be the counter that you work on, your kitchen counter, maybe the bottom of your shoe or the sink or doorknobs. You could even do the toilet seat, but you wouldn't be careful with that because there are possibly BSL-2 pathogens in the restroom. Maybe do the toilet after you've disinfected it. That would be an interesting variable to add to your hypothesis. But you want to pick 2 things and you want to formulate a hypothesis regarding which of these different inanimate objects, which of these fomites, do you think are going to have either more abundant bacteria, so more bacteria overall, or more species of bacteria, so more different colony morphologies growing on the plate, right? And then you want to have some kind of rationale as to why you have made that prediction, right? So a hypothesis has to have a predicted outcome with some kind of background knowledge, some kind of rationale, and you've got to be able to test it. So you're going to test it by, okay, you've picked your 2 fomites. You're going to denote one as being A, one as being B. Make sure you write down which one is which. Then you're going to prepare a nutrient agar plate. And again, if you've already pre-made these, then that's great. A pre-made plate, though, when you take it out of the fridge, may have a bit of condensation in there. If it's not a lot of condensation, that's totally fine. If you have a ton of it, you might kind of want to just pour some of that out into the sink before you use it. Condensation is normal, and if the plate is sterile, the condensation is also sterile, so you don't have to stress too hard about that. So you have your prepared nutrient agar plate ready to go, and just like last time, on the bottom you're going to divide. So this is the agar surface. So on the bottom of this, underneath here, you would draw with a Sharpie line down the middle and put one side A, one side B. And again, you're going to be using your sterile swabs and sterilized water. And you'll swab object A and use that swab to inoculate the A side with a simple streak pattern, and swab object B to inoculate the B side. Don't forget, when you're using these swabs, to roll the swab so you don't tear through the agar. You're going to tape the lid closed, just one little piece on one side and one on the other, just like we saw before in Lab 1. As you've seen already, and also throughout the rest of this course, we're going to be culturing and we're going to be growing bacteria. And it may seem kind of counterintuitive to want to grow bacteria because you're used to wanting to kill it. But microbiologists, in order to study microbes, you have to first grow them out. And in order to do that, you must be mindful of their specific pH, temperature, and oxygen requirements. Most bacteria are neutrophiles, which means that they grow at a neutral pH of about 7. Microbes that grow optimally at pH less than about 5.5, these are called acidophiles. At the other end of that spectrum are alkalophiles, and these are microbes that grow best at a pH between 8 and 10.5. Organisms that are categorized as mesophiles are adapted to moderate temperatures. So here we got temperature, rate of growth, mesophiles right here. Mesophiles will grow optimally from about 20 degrees Celsius to about 45. All of your human pathogens are going to be mesophiles because humans' temperature is about 37 degrees Celsius, which is right where mesophiles peak. Organisms called psychotrophs are those that prefer cooler environments. So from a high of about 25 degrees Celsius to about 4 degrees Celsius. So they like the lower temperature range. And then you have your thermophiles and hyperthermophiles. These are microbes that live in hot springs, geothermal soils, and things like garden compost heaps, where microbes are breaking down kitchen scraps, that kind of thing. These grow at very high temperatures. And some of your hyperthermophiles can even survive sterilization techniques like autoclaving, so they're very difficult to kill. Or at least they're very difficult to kill with high heat. Growth of bacteria with varying oxygen requirements is kind of shown in this figure here. In this tube here, you have your obligate aerobes. So they're not able to grow down towards the bottom where there's less oxygen. They can only grow up here where they have exposure to oxygen. The opposite of that is going to be your obligate anaerobes, where they can't grow anywhere where oxygen is perforating the surface, where it's in here. They can only grow down here at the bottom. Facultative anaerobes thrive in the presence of oxygen, but they can also grow in its absence. So in this case, you're going to have more growth towards the top, but also growth throughout. The aerotolerant anaerobes right here, they are basically indifferent to the presence or absence of oxygen. They don't use oxygen because they have a fermentative metabolism, but they're also not harmed by oxygen, so they do just fine with or without it, and they grow all basically evenly dispersed throughout the media. And then this guy here on the right, your microaerophiles. This is basically a Goldilocks situation because microaerophiles need oxygen, but they can't have too much oxygen. So the oxygen level has to be just right for growth, and they require a minimum level about 1 to 10% oxygen, whereas the atmospheric oxygen is about 20 to 21%. So microaerophiles are actually quite difficult to grow in the lab. Your microbes are going to need a constant nutrient supply in order to grow in the lab. So if you are running out of nutrients in the media, or if the temperature changes suddenly or anything like that, your microbes can die out. In nature, they would be getting their nutrients and everything from their environment, but in the lab, you have to provide everything artificially. To grow microbes in the lab, we use media, which is also called artificial culture media. The media contains the chemicals and nutrients for microbes that they need to grow. Media usually comes in a liquid form called a broth, or solid forms. Solid forms are going to be agar plates or slants or deeps. Liquid broth is very useful for culturing bacterial specimen quickly. That is, the incubation is usually 24 hours or less, so it's faster than growing on a plate. And that is a pro, right? But a con of that situation is that the bacteria will deplete the nutrients in a broth more quickly than they would in an agar. So it grows fast and it also dies fast. And you can tell that you have growth in a broth culture because the liquid will become cloudy, and the term for that is turbidity. If a broth is turbid, it is cloudy with microbial growth. And remember that broths are great for culturing, but they can't be used for isolation. So this here shows two different broths. This one has a bit more turbidity. It's a bit cloudy. It also shows how there has been a depletion of the nutrients by the microbes that are growing in it. And you can actually see kind of a layer of growth here on the top. This is a new sterile broth that doesn't have any microbes growing in it. It's nice and clear, and you can just kind of see by the color that the nutrients have not been depleted. Next you have agar. Agar comes in different forms, but the word itself, agar, it comes from an agent that's found in seaweed, and it doesn't provide any nutrients, but what it does is it solidifies the media. So you can literally have a broth, you can have a mixture of nutrients and chemicals, and it's in a broth form, and then you add agar to that, and the agar just solidifies it. So anytime you see agar, whether it's an agar plate or an agar slant or agar deep, you know that that is a solid media because it has this solidifying agent called agar in it. Then you usually have plated media that they're typically used for attaining isolation of species. We'll talk about isolation in a bit. They're also used for differential testing and quantifying bacterial densities. Two other types of solid media are known as slants and deeps. Slants are so named because the agar is actually solidified at an angle like this. So it's got a lot of surface area and a lot of exposure to oxygen. Deeps here are solidified straight up and down, and they have very little surface area and very little exposure to oxygen, especially towards the bottom of the tube here. These are inoculated in a stabbing motion, whereas slants you do very similar to a simple streak just on the surface of a slant, like back and forth on the surface like this. Whereas a deep, you actually stab it. So you can see here where it's been stabbed, and there's bacteria growing where it was stabbed. And the microbes are incubated inside the agar here, whereas on a plate or a slant, the microbes are incubated on the surface of the agar. These are actually placed inside of the agar, and there's much less exposure to oxygen. And because of the way that you inoculate these, you can actually test your microbe for motility. That is, you can observe whether or not the bacteria or the microbes were able to move away from the inoculation point in order to access either more oxygen or more nutrients or whatever. In other words, you can use a deep to see if your specimen have flagella or cilia or some other mechanism for movement. Because microorganisms are ubiquitous throughout the environment, it's very important for students to learn early in this course how to use proper aseptic technique. You do already know about wearing PPE, but in addition to that, aseptic technique involves avoiding contamination of your sample, your workspace, yourself, others. And one very easy thing to do is to make sure that you keep all of your specimen and agar plates and things like that at an arm's length. And you want to keep things covered, keep everything covered as much as possible. You always want to make sure that you're not having things too close to your face, you're not breathing into your plates, don't sneeze or cough while doing your lab procedures. And if you can't help it, then you can sneeze into your arm or wear a face mask. Another aspect of using aseptic technique is using sterile media and sterile equipment. So in a traditional lab setting, your instruments would be sterilized before and after use with either a Bunsen burner or a bactincinerator. So this here is a bactincinerator. This is the base and this is the heating element. And then this would be a metal inoculating loop. And we use these in my on-site labs instead of a Bunsen burner because Bunsen burners have those really big flames, the big open flames, and we use bactincinerators instead of those in order to reduce the risk of fire. You do still need to know how a bactincinerator is used, even though you're not going to be using these at home. It's still important that you know how it is used in a traditional lab. So how it's used is you would insert a metal loop into the heating element. You want to avoid scraping the sides. So you just push the loop in there towards the back and try not to scrape the sides. And then you hold that loop in the heating element for at least 5 seconds, but you don't want to hold it in there longer than about 10 seconds because you can actually melt the loop. These bactincinerators get up to about 400 degrees Celsius, so you can melt a metal loop in there. So about 5 seconds, no more than 10 seconds. And then once you take it out of the heating element, you want to allow it to cool. And you would not want to cool it by blowing on it. You don't want to set it on the counter because then it wouldn't be sterile anymore. So you just, basically the easiest way to cool a sterile loop would be to use sterile media, touch it to sterile agar, and if it sizzles, then it's not ready. If it's still sizzling, it's too hot. But you do want to cool it because if you cool the loop, then that prevents it from killing the bacteria that you're trying to grow. So if you want to use the loop to transfer bacterial cells from one place to another place in order to grow it, well, you don't want the loop to be so hot that it kills that bacteria. For the online labs, though, you're going to be using disposable loops instead of a metal loop and bactincinerator. So they're going to look something like this. These loops are made out of plastic and they've been sterilized before you get them. They'll be in like little pouches. And as long as you keep that pouch zipped up when you're not using it, you should be good. They'll stay sterile. You use one and then you dispose of it immediately. And in order to dispose of them properly, you're either going to put them in the hard plastic bins, the sharps containers that maybe you made for the last lab. You could put it in that, something with hard plastic sides. Or you could even just put them in a Ziploc bag, put them in the trash that way. That would be fine too. Whenever you're transferring bacteria, you want to hold the loop kind of like a pencil. So that allows your other fingers to be free to hold other things. Because a big part of aseptic technique is kind of learning how to hold more than one thing in your hands. Because you don't want to set things on the counter. Like you don't want to open your plate and put the lid on the counter because then your lid will be contaminated. Hold your loop like a pencil. It helps to kind of control where it's going, but it also allows your other fingers of your hand to be free to hold maybe a tube cap or something like that. And that way you're not contaminating things by setting them on the counter. For experiment 2, we're going to be learning how to perform proper aseptic technique while working with different types of media, and also again being able to observe colony morphologies. And you're going to be working with a Saccharomyces yeast. You're going to want to set this lab up ahead of time because you first have to culture the yeast in a broth, and then you're going to transfer it from a broth to a plate and culture it again. And that's going to take about 3 days altogether. So you want to set this up ahead of time. You're going to pour about half of your yeast powder, the yeast powder that comes in your kit. You're going to put that into the Falcon tube, which is also in your kit, and add about 10 milliliters of sterile water to that tube. You're going to allow that to incubate at room temperature for 24 hours prior to use. And you want to make sure that the lid of that tube you want it on there, but you want it to be kind of loose so that oxygen could get in there while the yeast is incubating. Then you're going to prepare a nutrient agar plate, and you're going to use one of the sterile disposable loops to obtain a sample of yeast from your broth culture. Basically, you're just going to dip the sterile loop in the broth. And when you pull the loop out, you should have a loop full of broth that has your yeast cells in it. And you're going to use that loop in order to inoculate your nutrient agar plate. And you're just, again, going to use a simple streaking pattern on the nutrient agar. And you're going to incubate that for 48 hours at room temperature. And while you are inoculating this plate, be very mindful of your aseptic technique. Make sure you're keeping it at arm's length. Make sure you're keeping the plate covered as much as possible. And what you're going to be looking for afterwards is you're going to make sure first you only have one colony type, because this should be a pure culture. You shouldn't have any contamination. And you're also going to be describing that colony morphology. So we'll talk about that some more in a bit. So speaking of having only one colony type, if you have a mixed culture, which a lot of times you will, because in nature, bacteria live in mixed cultures. They don't just hang out with their own kind. They're all swimming around together, right? So you have a mixed culture that has more than one species in it. And in order to get a pure culture that has only one species in it, you're going to have to first isolate that specimen. So what you're doing is you're taking a mixed culture or even a pure culture. You can always isolate another pure culture. But the point is that you are going to use a very specific technique in order to obtain isolated colonies. And a colony, you've heard me say the word a few times now, term itself refers to a single cell that has grown and reproduced in such a way that you can observe it, like you can observe this aggregation of cells on a plate with the naked eye. But every cell in that aggregation is genetically identical to each other because they all derived from one single cell that divided and reproduced asexually. The technique itself for doing this, for obtaining individual colonies of pure cultures, is called the streak plate technique, or streaking for isolation. Streaking for isolation consists of using a sterile loop to inoculate an agar plate using a primary streak followed by a second, third, and fourth streak, and sterilizing, or in your case, you're going to be switching out the loops between each streak. The inoculum is obtained, and then you streak it into the first quadrant right here. So you get your inoculum, streak it, just a little simple streak, but only in one quadrant of the plate. Then you sterilize the loop, or in the case that you're using disposable loops, you would dispose of the first loop and get you a new one. And then you're going to pull just a little bit from the first quadrant into the second quadrant and streak it out. And then if you're using a metal loop, you would sterilize it. If you're using a disposable loop, you would dispose of that loop and get a new one. And then pull a little bit from the second quadrant and streak it out into the third. And then one more time, get you another sterilized loop, and then pull a little bit out from the third into the fourth. So what's happening here is there is a dilution of the cells from one quadrant to the next. And eventually this is hopefully going to lead to you having individual cells that are deposited onto the agar surface. It usually happens in the fourth quadrant, sometimes the third quadrant. But you're starting with tons and tons and tons of cells here, like tens or hundreds of thousands of cells that you're pulling from the broth. And then you're physically diluting that out until hopefully somewhere on the plate you'll have one cell, one cell, one cell. And then those single cells will propagate until you will have pure isolated colonies on the plate. So this is what they tend to look like in real life. So each one of these little dots that's by themselves is an individual colony, and those individual colonies derived from single cells. So you know that they are 100% pure culture. Experiment 3, you're going to be doing a simulation. And this is because we have a BSL-1 limitation for the online lab. I couldn't send E. coli and M. luteus or other organisms home with you, so what you're going to be doing instead is a simulation. It's a streak plate simulation where you will get to do the technique virtually, and it's found here at this URL. And in fact, I've got a little short clip here for you, kind of showing you what that will look like. So you're going to want to do that simulation, and you want to observe and record all of the procedural steps and the results so that you'll have them for completing your lab report. So very similar to Lab 1, you want to make sure that you keep your lab specimen and reagents away from any food or drinks. You're going to disinfect your counter or surface wherever you're working before and after performing lab procedures. If you have access to PPE, I highly recommend using PPE, especially when you're working with any live specimen like your yeast culture or your Ubiquity plate. Be sure to carefully read and follow the lab procedures that are provided. Treat all specimen as potential pathogens. Make sure you decontaminate your plates and broths and all of that stuff before you dispose of them. And don't use your counters or surfaces for preparing food for at least an hour after you've decontaminated it. As for observations and interpretations for experiment 1, again, you're looking to see if your hypothesis was supported or rejected. And you're going to be looking at either the abundance of growth or the numbers of species on A versus B in order to see if your hypothesis was supported or rejected. For experiment 2, you're going to look at the density of the growth in the broth, and you're going to look at the density of the growth on the plate. You also are going to do a little bit of literary research because I want you to look up and see what the oxygen requirements are for Saccharomyces and if you should have expected it to grow well in the broth or the plate. And then for the plate culture, the simple streak, you want to have a look at the growth patterns and the morphologies to see if you actually did obtain a pure culture of yeast on your nutrient agar plate, or if you perhaps had some contamination and need to work on improving your aseptic technique. For experiment 3, you're going to be talking about whether or not you have successful isolation of colonies. And you're going to discuss why it was so important to sterilize the loop between quadrants, why it's necessary to cool the loop between quadrants. And again, here are some of the terminologies and things you're looking for when you're describing colony types, colony morphologies. You have things like shape, size, margin, color, elevation. This is an example of what you might see on your Ubiquity plate. And again, it kind of looks like just a whole bunch of dots on a plate, except it's not. You have some things that are semi-transparent. You have some things that are fully opaque. You have smooth margins. You have irregular shapes. You have things that are more of kind of a yellowish color, some more things that are much more of a white color. Here you have an umbonant colony, and you can tell because it's more raised in the middle, flat on the sides. There's another one there. This is a rhizoid. This, in fact, is probably a mold. Okay, so those are some things you look for the differences. You have things that are much larger here, and then things that are very, very tiny. This is a punctiform colony. So at this point, we've really got to get kind of used to describing the morphologies. This is an example of what a Saccharomyces yeast should look like if it's a pure culture. It's got a nice kind of off-white color to it. It's raised. It's pretty large. It's shiny, and shiny is a technical term. Shiny means that it reflects light, whereas dull means that it doesn't reflect light. And this is an example of what your streak plate should look like. It's a simulation, but it shows you what your result is after incubating. So you should have some isolated colonies, either in 3 or 4 at the end, and no contamination. You can tell if it's contaminated if you have growth that doesn't follow the streaking pattern. So maybe something fell in the plate. Or if you have growth, you have colonies that are not all the same. Because this came from a pure culture of E. coli, it should look like a pure culture at the end. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.